For the WBCHSE Board, we define excretion precisely. It is the biological process of eliminating metabolic waste products and toxic substances to maintain internal cellular homeostasis. All metabolism inherently generates chemical exhaust. If these toxic byproducts are allowed to accumulate, the internal cellular environment acidifies, chemical gradients collapse, and the cell dies of toxicity. This microscopic footage shows fluid accelerating through a highly active, branching biological network. Because animals maintain a mobile, high-energy lifestyle, they consume vast amounts of fuel and generate enormous amounts of waste. To survive, they require these continuous, high-speed biological transport systems to actively filter and flush toxins out of the body. Plants operate under different constraints. Being stationary, their energy demands and metabolic rates are comparatively low. This slower chemical turnover allows them to manage waste through passive, slow-acting processes rather than rapid filtration. Look at this matrix comparing the two systems. An animal's high metabolic rate on the left necessitates an active transport and filtration network. A plant's low metabolic rate on the right permits static cellular storage. To master this module, we will analyze the physical processes of plant elimination, categorize their specific excretory products, and trace the evolutionary development of active excretory organelles in animals. An organism's method for handling waste is a strict anatomical consequence of its energy expenditure and physical mobility. Plants completely lack a centralized excretory network. There are no kidneys, no specialized filtration organs, and no continuous fluid streams dedicated solely to waste removal. Instead, plants repurpose their waste. Through internal osmoregulation, these chemical byproducts are locked away to maintain cellular pressure, and they frequently serve as toxic chemical defenses to deter grazing herbivores. To physically eliminate these stored toxins, plants rely on three specific architectural mechanisms. The first is abscission, or leaf fall. Plants actively transport metabolic toxins into the large vacuoles of aging leaf cells. The leaf acts as a disposable storage container. Once it reaches capacity, the plant seals off the stem, and the yellowed leaf detaches, taking the accumulated waste with it. The second mechanism is bark shedding, a primary strategy for heavily timbered trees like the eucalyptus. As the tree grows, it continuously pushes complex chemical wastes outward into the older, dead bark tissues at the perimeter. When the bark eventually dries and peels away from the living trunk, the trapped waste is discarded. The third method is fruit drop, utilized extensively by apple, lemon, and tamarind trees. Specific organic acids, like malic acid or tartaric acid, are aggressively sequestered within the tissues of developing fruits. This removes the harsh acids from the plant's main circulatory system and stores them safely until the ripe fruit falls. Lacking a system for continuous excretion, the plant relies entirely on a strategy of localized chemical isolation followed by controlled tissue amputation. This flowchart categorizes plant wastes by their chemical composition. The system divides strictly into nitrogenous wastes, specifically alkaloids, and non-nitrogenous wastes. Alkaloids are complex organic compounds characterized by the presence of nitrogen atoms. Assembling these nitrogen-heavy molecules costs the plant significant biological energy. The investment pays off because alkaloids are highly bitter and neurologically toxic to the herbivores that try to eat them. The first crucial alkaloid is quinine. It is extracted directly from the bark of the cinchona tree and in human medicine, it functions as a highly effective anti-malarial drug. Next is reserpine, sourced from the roots of Rauwolfia serpentina, or sarpaganda. Clinically, it acts as a powerful agent for lowering high blood pressure. Datura, or stramonium, is sequestered in the leaves and seeds of the datura plant. Medicinally, it functions to relieve severe asthma and respiratory spasms. Finally, caffeine is accumulated in the beans of the coffee plant and the leaves of the tea plant. In nature, it is a lethal insecticide. For humans, it acts as a central nervous system stimulant. For your exam, you must be able to strictly pair each of these specific biological source tissues with their corresponding functional alkaloid. These alkaloids operate as defensive biochemical weapons for the plant. Human pharmacology has simply mapped and extracted them to use as critical medicines. Returning to the chart, we isolate the non-nitrogenous wastes. These are the direct metabolic byproducts of carbohydrate breakdown. Lacking nitrogen, these compounds do not act as chemical toxins. Instead, their physical properties provide structural sealing and mechanical defense. Resins are sourced from the stems of coniferous trees like pine. The plant uses them to seal physical wounds against infection. Humans harvest them to manufacture varnishes and polishes. Gums are exuded from the stems of acacia and neem trees. They function similarly in wound healing for the plant and provide the raw material for commercial adhesives. Latex is a thick, milky fluid stored in special cells of the rubber tree and papaya plant. It offers internal tissue protection and forms the absolute basis of global industrial rubber manufacturing. Tannins are stored in tea leaves and myrobalan. They deter infections and are harvested heavily for the commercial tanning of animal leather. The thick, viscous properties that allow a plant to seal a severed branch are the exact properties that make these substances industrially valuable for varnishes, rubbers, and glues. Non-nitrogenous wastes act as a biological recycling loop, converting the exhaust of carbohydrate metabolism into structural armor. We return to the animal kingdom. Because their high metabolic rates prevent them from passively storing toxic exhaust, they require dedicated active hardware to push waste out of the body. As anatomical complexity increased across species, the hardware required to filter waste upgraded sequentially. We can map this progression on this phylogenetic ladder. At the baseline, single-celled amoeba and protozoa rely on simple diffusion across the plasma membrane, utilizing contractile vacuoles strictly to pump out excess water and regulate osmotic pressure. Stepping up to the flatworms, such as planaria, we see the first specialized excretory organelles, flame cells, also known as protonephridia, which actively filter internal fluids. In annelids, like the common earthworm, the system upgrades to nephridia, coiled tubular structures that extract waste from the body cavity. In arthropods, specifically insects like the cockroach, waste management is handled by Malpighian tubules, which extract uric acid and deposit it into the digestive tract to conserve water. You must note two specific arthropod variants. Crustaceans, like prawns, rely on green glands. Arachnids, like spiders, rely on coxal glands. Reaching the top of the ladder with vertebrates, including fish, birds, and humans, we find the kidneys, macro organs driven by a highly complex network of microscopic nephrons performing continuous blood filtration. The evolutionary record of animals tracks a direct line from basic membrane diffusion to highly engineered, pressure-driven filtration organs. Viewing the complete plant flowchart alongside the animal phylogenetic ladder clarifies the core dichotomy in how life handles metabolic exhaust. Plants rely on defensive biochemistry and structural tissue shedding. Animals rely on anatomical engineering and continuous fluid filtration. To secure top marks on your exam, verify 3 areas of knowledge. First, cleanly distinguish between toxic nitrogenous alkaloids and structural non-nitrogenous wastes. Second, memorize the exact biological source tissues for every plant product. Know which alkaloid comes from bark, which comes from roots, and which from seeds. Third, accurately match every animal phylum discussed to its specific excretory organelle, from flame cells and flatworms to Malpighian tubules and insects. Whether it is a tree shedding a leaf or a human kidney filtering blood, the biological goal remains identical: preserving internal cellular homeostasis. Biology wastes nothing. The chemical exhaust that would poison a cell is predictably harnessed, becoming the defensive armor of a tree, the medicine that cures disease, and the evolutionary pressure that built our own internal organs.